Hello everybody, welcome to my course, How to Remember the Vocabulary You Learn. Overview. This course is meant for all those who are appearing for the GRE, SAT, TOEFL, IELTS exams and for all those who wish to improve their English comprehension skills. Answer to riddle number 4. Do you remember what the question was? I'm used when you jump off a bridge for fun. What am I? The answer was bungee jumping. Have you tried it? Well, I would like to try it. I haven't yet done it. Anyway, moving forward on that adrenaline kick. Lesson 5 deals with neuroscientists plant false memories in the brain. MIT study also pinpoints where the brain stores memory traces, both false and authentic. This is written by Anne Trafton, MIT News Office. I will be reading this article on false memory. Pay attention to what I'm reading. This will not cover the vocabulary, but in the end you will have comprehension self-study questions. So let me start reading. The phenomenon of false memory has been well documented. In many court cases, defendants have been found guilty based on testimony from witnesses and victims who were sure of their recollections, but DNA evidence later overturned the conviction. In a step towards understanding how these faulty memories arise, MIT neuroscientists have shown that they can plant false memories in the brains of mice. They also found that many of the neurological traces of these memories are identical in nature to those of authentic memories. Whether it's a false or genuine memory, the brain's neural mechanism underlying the recall of the memory is the same, says Susumu Tonegawa, the Pi Cover Professor of Biology and Neuroscience and senior author of a paper describing the findings in the July 25 edition of Science. The study also provides further evidence that memories are stored in networks of neurons that form memory traces for each experience we have, a phenomenon that Tonegawa's lab first demonstrated last year. Neuroscientists have long sought the location of these memory traces, also called engrams. In the pair of studies, Tonegawa and colleagues at MIT's Pi Cover Institute for Learning and Memory showed that they could identify the cells that make up part of an engram for a specific memory and reactivate it using a technology called optogenetics. Lead authors of the paper are graduate student Steve Ramirez and research scientist Zhu Liu. Other authors are technical assistant Pei Ann Lin, research scientist Jung Yup Su, and postdocs Michelle Pignatelli, Roger Redondo, and Thomas Ryan. Seeking the engram. Episodic memories, memories of experiences, are made of associations of several elements, including objects, space, and time. These associations are encoded by chemical and physical changes in neurons, as well as by modifications to the connections between the neurons. Where these engrams reside in the brain has been a long-standing question in neuroscience. Is the information spread out in various parts of the brain, or is there a particular area of the brain in which this type of memory is stored? This has been a very fundamental question, Tonegawa says. In the 1940s, Canadian neurosurgeon Wilder Penfield suggested that episodic memories are located in the brain's temporal lobe. When Penfield electrically stimulated cells in the temporal lobes of patients who were about to undergo surgery to treat epileptic seizures, the patients reported that specific memories popped into mind. Later, studies of the amnesiac patient, known as HM, confirmed that the temporal lobe, including the area known as the hippocampus, is critical for forming episodic memories. However, these studies did not prove that engrams are actually stored in the hippocampus. Tonegawa says, to make that case, scientists needed to show that activating specific groups of hippocampal cells is sufficient to produce and recall memories. To achieve that, Tonegawa's lab turned to optogenetics, a new technology that allows cells to be selectively turned on or off using light. For this pair of studies, the researchers engineered mouse hippocampal cells to express the gene for channel rhodopsin, a protein that activates neurons when stimulated by light. They also modified the gene so that channel rhodopsin would be produced whenever the C-force gene necessary for memory formation was turned on. In last year's study, the researchers conditioned these mice to fear a particular chamber by delivering a mild electric shock. As this memory was formed, the C-force gene was turned on, along with the engineered channel rhodopsin gene. This way, cells encoding the memory trace were labeled with light-sensitive proteins. The next day, when the mice were put in a different chamber they had never seen before, they behaved normally. However, when the researchers delivered a pulse of light to the hippocampus, stimulating the memory cells labeled with channel rhodopsin, the mice froze in fear as the previous day's memory was reactivated. Compared to most studies that treat the brain as a black box while trying to access it from the outside in, this is like we are trying to study the brain from the inside out, Liu says. The technology we developed for this study allows us to find, dissect, and even potentially tinker with the memory process by directly controlling the brain cells, incepting false memories. The researchers then showed that immediately after recall of the false memory, levels of neural activity were also elevated in the amygdala, a fear center in the brain that receives memory information from the hippocampus, just as they are when the mice recall a genuine memory. The MIT team is now planning further studies of how memories can be distorted in the brain. Now that we can reactivate and change the contents of memories in the brain, we can begin asking questions that were once the realm of philosophy. Ramirez says, are there multiple conditions that lead to the formation of false memories? Can false memories for both pleasurable and aversive events be artificially created? What about false memories for more than just contexts? False memories for objects, food, or other mice? These are the once seemingly sci-fi questions that can now be experimentally tackled in the lab. The research was funded by the Riken Brain Science Institute. Well, we come to the Comprehension SelfStudy. There are 3 tricks to learning vocabulary. One was to learn the opposites of the word that we're going to discuss. 2 was to practice and build the words. 3rd was to use your imagination. But in this article, most of the words are self-explanatory. So I thought it would be a good idea to just go directly to the Comprehension Self-Study. You could probably check out your answers with your friend or maybe do it individually at your own pace and try to understand what this article was all about. The first question was, what did the MIT study on mice teach scientists about memory formation? 2. The unreliability of memory is a serious problem when it comes to witness testimony. Why? 3rd one, explain the potential effects the study of memory formation could have using at least one example from the text. So go ahead and answer this. You can probably check out your answers by reading the paragraph once again. And we come to the last part of this lesson, riddle number 5. Why did Adele cross the road? Answer, to sing hello from the other side. I found this very funny. I hope you did too. This brings us to end of this course on how to remember the vocabulary you learn. Stay alert, keep learning, keep shining, keep smiling. Thank you for watching this. Share this with your friends. Do write to me with your comments, suggestions and feedback. I'd love to hear from you all. Bye for now.